Good evening. I'm BJ White. I will be the moderator for this evening's community forum entitled Demystifying Cannabis and Hemp. Thank you for your interest in joining this Zoom meeting on a topic that is of growing interest to many. There are a few housekeeping instructions to discuss before I ask Shirley Schultz to introduce our speaker. You will be muted unless you are selected to ask a question. Number two, you are free to listen and see the speaker and his slides. But if you choose to have only your name and not your face on the screen, you simply click on video in lower left side of your screen. Questions and answers will follow the presentation. Click on the chat button if you want to type your question, or click on the reactions button and then the raise your hand button to get into the line to ask your question. When and if you are selected, the moderator will recognize you so you can ask your question. Please keep your questions of a general nature that might be of interest to everyone, and avoid giving details of your personal histories. Thank you. And now Shirley Schultz will introduce tonight's speaker. Shirley.
Good evening, everybody. It's so nice to see all of you. This is the first time I've participated in a Zoom community forum, and it is truly my honor this evening to introduce Dr. Jehan Marcu, PhD. He's a researcher and an international speaker, very qualified to speak to us about medical uses of cannabis, its production and its safety. Dr. Marcu is currently the Chief Operations Officer, Director of Experimental Pharmacology and Behavioral Research at the International Research Center on Cannabis and Health, which he co-founded. Formerly, Dr. Marcu was the Chief Science Officer at Americans for Safe Access, and also was the former director of its patient-focused certification program, a health and safety oversight program that assesses regulatory compliance at cannabis operations. Dr. Marcu published the first direct measurements of THC and CBD synergy, for which he received the Billy Martin Research Award from the International Cannabinoid Research Society. He has developed pharmacogenomics, that's a tongue twister, testing technology used commercially and in research studies to predict drug-drug interactions with cannabis, as well as using the technology to guide dosing and administration forms for patients. You can read articles by or view videos of Dr. Jehan Marcu by going to the website for his International Research Center on Cannabis and Health. Since Dr. Marcu is speaking to us from New York tonight without any fee or reimbursement, I'm sure he would appreciate your donations, which he can be made on the IRCCH website. The most recent article he co-authored was published in the August 31st, 2021 Archives of Rheumatology and Arthritis Research, titled A New Ache in the World of Joints. Finally, I want to tell you how it is that we have this special speaker this evening. In early 2019, over 2.5 years ago, I was volunteering at a community event with Patty Jackson Brown, and she gave me permission to use her name tonight. And I was telling her about my interest as a member of the health education team in finding a speaker on the topic of medical marijuana. She told me about her grandson, Jehan Marcu, and put me in touch with him. He is set to speak with us. He was going to speak with us in March of 2020 before we were shut down by COVID-19. So without further ado, Dr. Jahan Marcu, and your grandmother is watching.
Oh, the pressure. Thank you so much. It's really a pleasure to finally be here. And just as a matter of housekeeping, you can all see this beautiful slide, correct?
Yes.
Yes.
All
Yeah.
right. So today, as you said, we'll be discussing a presentation entitled Demystifying Cannabis and Hemp. We're going to talk about pharmacology, safety, and some regulations. And one of the most important things you're going to learn tonight is actually on this slide. And that's my email address, because you're going to have questions. You might want copies of some of the studies I'll talk about. So, and don't worry, this will be email address will be shared at the end as well. But if you want to make a note of it, I think it's important, but we'll come back to that. So, I think it was a great job you did on introducing me, and I just wanted to give a little more color and shade in some things. A lot has changed in the last months, years, I don't know, however long it was since COVID happened and things shut down. But my PhD is in molecular pharmacology. I've spent 18 years working in academia, working on policy, and in the cannabis industry itself to some degree. My latest venture is a consulting firm called Marcu and Aurora, and we've been operating for about a year now. My PhD was on studying the structure and function of cannabinoid receptors, otherwise known as the target of THC and other compounds from the plant. I'm a basic and clinical researcher, and I'm going to share some data about that with you. One of the things I'm both known for, both loved and hated for, is helping create the first industry standards for cannabis and hemp, which include the testing standards, the basic safety testing parameters for lead and mold and things like that. I'm an editor-in-chief of the American Journal of Endocannabinoid Medicine, which I also helped start. I provide consultation to regulatory bodies such as the state of Delaware's Department of Health, as well as to industry. And I can't tell if that's a disclaimer or just bragging, but I'm also a court-qualified synthetic cannabinoid and cannabis expert. And if you ever want to have a Coca-Cola and hear about some amazing stories, I highly recommend you getting my time. I also serve as adjunct faculty at the University of Sciences, where I teach pharmacy and business students about cannabis, everything from supply chain to the pharmacology of cannabis. The photo on the left, as you can see, is a gratuitous picture of hemp. And that's when I was invited 2 years ago to China as a guest of the government to what they said, and I'll take their word for it, the first pharmacologist invited to speak on the activities of cannabis. And that's in the northern province of China in Harbin. So I love to start my official presentation with quotes. And because we're going to be talking about a lot of things, trying to open up the hood and look under, at the engine of cannabis and the system it acts upon. And we're going to end with reviewing a label of 2 products. I really love this quote from a research study we're going to discuss. And that is, my feeling is that every single cannabis product should have a standardized drug fact label required. Like when you read the back of a sunscreen label, you can understand it because the formatting is the same. And we're going to, I think that this is such a beautiful quote to summarize the state that the cannabis industry is in, a quilted patchwork of regulations that are not the same depending on where you are. So we're going to attempt a big feat today. Several subjects we'll attempt to get over. We're going to talk briefly about the history of cannabis, and we're going to spend a lot of time talking about pharmacology, mechanism of action. I'm going to spend some time talking about opioids in cannabis, a very hot topic right now, as well as sharing some of my clinical research in the area. I'm going to talk about drug-drug interactions, a fascinating topic, and helping to sort of balance out the therapeutic and the risks of these compounds. We'll also talk about labeling issues and quality, and talk, hopefully you'll leave here with a little bit of science and some applicable information to help guide you through this world. And remember, I'm going to use a lot of big words. Maybe some of it will be unfamiliar, but if it is important, I will repeat it many times. So the story of cannabis is an old one, and it begins in ancient times. Every culture that has written records makes a note to cannabis or hemp in some form. Some of the best records come from China, where cannabis was thought to evolve from. In their Materia Medica, the Emperor Chang Nung, who is the father of modern agriculture, wrote extensively about cannabis, mostly in the realm of for women's health issues. It's in the Atharva Veda text in India, all over there. It's actually Shiva's favorite plant. There's a reason Shiva is blue and the eyes are squinted open. Um, Cannabis concoctions such as bong or bang are religious sacraments in other parts of the world. The first evidence of cannabis to treat seizure disorders actually comes from documentation in the Arab world. And it wasn't really until the 1850s that the Western world learned of the bioactivity of cannabis. Cannabis is actually, or hemp or marijuana, is not indigenous to the United States. It was brought here by two different mechanisms. Um, some of you may be familiar with the colonists. They brought over hemp because it was a very useful fabric, especially for making sails on ships and things like that. And Cortez also brought it in with him with the Spanish on the other side of the continent. And every missions throughout the western part of the United States, as well as Mexico, grew hemp. And if you go to San Francisco to the mission district, to an old mission, they actually had a hemp plantation there at one point. But some of the things I'd like to draw your attention to include that THC in a synthesized form was actually a Schedule III drug available by prescription in 1985. And if you look at the bottom timeline, we actually didn't even discover the receptors in the scientific community until the 1990s. We didn't know about this endogenous system until the mid-90s. So 10 years before we knew how the drug worked, it was approved for HIV, AIDS, wasting, and cancer-related nausea and vomiting due to chemotherapy agents. Some of the biggest advancements have come from the approval of Epidiolex, which is a CBD product, which we will discuss briefly. And I'd love to come back and just do a history talk. So I could talk about this all night. The history is quite fascinating, including the references in the Bible to cannabis and its use in anointing oils and things like that. So cannabidiol is really CBD, hemp CBD, however you want to call it, has had a meteoric rise. And it really began with a company breeding for this product in the 1980s. In the 1996, Prop 215 was passed. That is the measure in California, which its anniversary is coming up in November. Then the endocannabinoid system was discovered after the medical marijuana program was initially started. And GW Pharma, who created Epidiolex and other products and cannabis products recently purchased by Jazz Pharmaceuticals, acquired Hortifarm's CBD plants. And this was at a time in the early 2000s when there were maybe 8 dispensaries in the entire country. And they were killing CBD plants. Nobody wanted it. And Project CBD began, a group that I provide content for, began actually trying to keep the plants alive. And shortly after that, the Stanley brothers came on the scene, rebranded themselves, and started producing CBD products from plants that were basically given away. Nobody wanted it, saw the value in it. I remember the first time I went into a dispensary and talked about CBD, and they said, well, what does it do? I said, well, it doesn't get you high, and it actually blocks the intoxicating properties of THC. And they said, that sounds terrible. Who would want that? And then, so over the next 10 years, 15 years or so, we would see the FDA grant emergency status to this in terms of getting it through the FDA pipeline quickly. And that also paralleled this confusing thing called the Hemp Farm Bill, of which we've had 2 of, which created a false dichotomy, which I think has created a lot of confusion about THC and CBD. Hemp and cannabis are the same plant. Hemp is now a regulatory definition where the plant material has to have 0.3% or less THC in the United States to be called hemp. But this has led to a number of issues I think you might be familiar with if you've heard about Delta-8 THC and things like that. And we'll discuss this issue a little bit later. But 2 of the most promising molecules, and I'll go into more details in these later, are THC and CBD. Now, THC was first isolated in the 1960s. CBD was actually isolated first in the 1950s by an American chemist, Roger Adams, of which the American Chemical Society has an award for. Everyone likes to give Israel credit for everything, but many discoveries are actually made in the United States, including the discovery of the receptors, the endocannabinoid system, as well as the first purification of a cannabinoid in 1890 was conducted by an American chemist. So the cannabis plant produces these compounds, among which are THC, and it stimulates the CB1 and CB2 receptor, cannabinoid receptors, which I'm going to go into a lot of detail in a minute. Cannabidiol, I want to address this right away, does not stimulate cannabinoid receptors. And we're going to spend a little time talking about its special interaction at the type 1 receptor. But CBD affects transient receptor potential vanilloid type 1 receptors, or TRPV1 receptors, as we like to call them. It also can interact with serotonin receptors, 5-HT1A. Do not get that confused with 5-HT2A, because if you stimulate a 5-HT2A, you will have a hallucination. That is the target of psilocybin and other hallucinogens. But the 5-HT1A receptors is also a target of a lot of anti-anxiety medication. And CBD also interacts with the A2A receptor, the adenosine receptor. This is the receptor for one of my favorite drugs, probably my favorite drug, and I have some of it here. It's some tea, caffeine, nothing like a little adenosine stimulation to get you through a presentation. So cannabis has thousands of varietal names. Silver Haze, White Widow, Tropic Thunder. I don't know, make it up. It probably exists. But there's actually only a few chemical varieties or chemovars, THC predominant, CBD predominant, or a mix of the two. And whether or not it's isolated from hemp or cannabis, these molecules have the same pharmacology. So where do these come from exactly? So from a book chapter I wrote with my colleague, Dr. Ethan Russo, just simply, you start with a cannabis plant. Some people say, oh, God gave us this plant. Actually, it's isolated from trichomes. So when you look at this plant, you might think, well, what part of the plant is useful? Well, if you're looking to isolate THC and CBD, you're not going to find it in the roots, but you will find some very interesting alkaloids and terpenoids, these fragrant compounds that have some medical utility. And we'll touch upon some of these interesting compounds as well. You also have the stem or the stalk, which is typically retted, that is rotted, and shucked for its paper and textile use. BMW's electric car, actually a lot of the paneling that looks like wood is made from pressed hemp. The fan leaves of the cannabis plant, as popular and iconic as they are, you don't find many cannabinoids there. Where you find the oil of the plant is in the unfertilized flower in a very particular type of trichomes. Now, plants have trichomes not for our pleasure, but for their defense and reproduction and for sending chemical signals. And it's in targeting these glandular trichomes and unfertilized flower tops is where you get the oil from. So no matter what someone tells you, there should not be any cannabinoids in seeds or seed sprouts, very little in the stem. The only reason there would be drug material in seeds is if someone was very, very sloppy and acquiring the seeds through a resinous plant. But again, the seeds do not contain any cannabinoids. They're actually very nutritional, one of the most nutritionally dense seeds, I believe second only to sunflower seeds. So why do we care about this specificity, about THC and CBD, about this plant? Because it led to the discovery of the endocannabinoid system. This system is an adaptive system that responds to stress. It is a homeostatic regulator. It helps to regulate homeostasis. Homeostasis is a great thing. It's also maybe not so great because sometimes, homeostasis is the reason why you have to keep doing things to learn them. Your body wants to keep you in a certain alignment, but it's also why your brain isn't just distracted by every little thing that's out there in the world, by every little stray thought, every little squirrel that runs by. There is an endogenous system. The reason that cannabis has an effect on us is because we have a receptor system similar to endorphins, which is just a shorthand word for endogenous morphine. So we have pain relieving compounds that are produced. We also have endogenous cannabinoids, and these govern a lot of physiological processes. What do they do? What does this process of receptors and endogenous ligands do? It helps us to eat, sleep, relax, forget, and protect our body from harm. So some of you may be familiar from watching movies and the popular literature, stimulating appetite, sedation, sleep, relaxing. But one of the most important aspects to me, and one of the most interesting, is forgetting. If I remembered every face I saw on, on the New York subway, my head would explode. You need a system in your body that helps you get rid of information that's no longer helpful, potentially harmful. And that's why we see a lot of drugs and treatments in the cannabis space targeting things like post-traumatic stress disorder, targeting effects like mood, and other issues. And this system also helps protect our body through harm by homeostatic regulation. So here's a fun little diagram. So this is a cross section of a politician's brain, and you can tell it's an accurate model of a politician's brain because there's only one neuron that is functioning. So what we're seeing here is a convergence on the cannabinoid receptor, whether it's THC from the cannabis plant or anandamide, which is the sort of THC your body naturally makes to carry out its business related to eating, sleeping, relaxing, forgetting, and protecting your body from disturbances to its homeostatic regulation. You also have another friend of anandamide, 2-AG. There's a whole family of endocannabinoids, and they mimic the same effect. Now, if you really want to impress a doctor friend of yours at a little tea party, you could say, well, cannabinoid stimulation, cannabinoid receptor stimulation simply leads to an inhibition of glutamate release. They will be very impressed because they probably did not learn that in medical school, as the system was only discovered in the late 90s and is still yet to be incorporated into the medical school curriculum in the United States. But there are some interesting and fascinating aspects about just this exquisite interaction that the system has. It's a retrograde signaling system. So what that means is that at the top here, we have what we might call the presynaptic neuron. And below that, the postsynaptic neuron. And if that's too wordy for you, we can call it A and B. And so from A, there is a excessive release of glutamate, a signal from the brain. Maybe it's a stress marker. Maybe it's leading to a seizure. Something is overexciting the neuron. Its buddy, neuron B, or the postsynaptic neuron, detects this. It starts synthesizing on demand endocannabinoids from the cell wall, which then go and shut off this excessive activity. And this protects neurons from exciting themselves to death. And this is a very important aspect and a very unique aspect of cannabinoids. Nitric oxide, which is used for vasodilation in certain parts of the body. That is one of the only other areas we see retrograde signaling in the human body. So there are a couple unique mechanisms here in regards to how these things function. So where can I find these useful cannabinoid receptors? That is a great question. So the CB1 receptor, the type 1 receptor, mostly associated with the effects of THC, is widespread, has a high level of expression, and is the most abundantly expressed protein in the mammalian system. That is to say, if you were to take opioid receptors, serotonin receptors, someone shout out another receptor, yeah, we'll add that one to it. There are still more cannabinoid receptors in your brain than just about any other protein. Receptors are proteins. We have to remember that. And one thing that's also interesting is that they are located everywhere, except in the brain stem. They're sparsely found there. And that is a fundamental importance and separates them from things like opioids, which are opioid receptors are found in the brain stem. And stimulating those leads to respiratory depression and oftentimes death. And we'll discuss some aspects of opioids later as well. And so cannabinoids aren't found in high levels in the brainstem. The brainstem also is from the reptilian brain. We have things like some fear is rooted there, greed, the willingness to follow leaders blindly, behaviors we can still see to this day. So when it comes to cannabinoid receptors, the distribution of these receptors parallels what you can see, right? It increases appetite. Well, you'll find receptors in the nucleus of the solitary tract, which is important for controlling appetite. Mood, memory, in the hypothalamus. Cannabis is often associated with jazz musicians, affecting motor response and improvisation. Well, you'll find it in the motor cortex, the basal ganglia, the cerebellum, as well as in the spinal cord. You find it in the eye, helping to control eye pressure, the immune system. You find it in cancer cell lines, as well as in the heart, lung, adrenal, and liver. And there's also the most important part of the brain, the Snoop Dogg region of the brain. And if you guys don't like these jokes, just let me know in the chat. Oops, skipped one too many there. All right, so the type 2 receptor, we have a type 1 located in neuronal tissue, the brain, spinal cord, and the nerves that pervade the system and send fun little signals to help you move your muscles. But you also have a type 2 receptor. And this is located in things like the bone marrow, thymus, spleen, tonsils, largely the immune system. One of the things that's really amazing about stimulating CB2 receptors is that it helps decrease inflammation. And part of my thesis work was on actually studying the role of this receptor system in bone, osteoclasts, osteoblasts, osteocytes, things that degrade bone, things that lay new bone, and the balance, the sort of yin and yang of these bone cells that help replace your skeleton every 8 to 10 years entirely. So type 2 receptors, mutations in these receptors are actually associated with osteoporosis. That is, we could speculate that a less functioning CB2 receptor, as been documented in mice, have accelerated age-related trabecular loss. So in 3 distinct human populations, in Japan, France, and in a small group of people living along the Volga River in Eastern Europe, type 2 CB cannabinoid receptors, polymorphisms, have been shown to be associated with basically not healthy bones later in age, more age-related bone loss. Now, while CB1 receptors are found in the brain, you do find CB2 receptors in the brain, but you have to be very specific. They're found on the immune cells of the brain, the microglia. Well, when it comes to where they actually are on cells, and this is something we published in the Journal of Biochemistry. For nerds, this was a major finding. Typically, when we think of cannabinoid or any receptor, we think of it on the outside of the cell, along the plasma membrane. And some of you may remember this figure from 8th grade biology. But you also have cannabinoid receptors in the cell that are being trafficked to the surface, and those can actually be stimulated as well and targeted. And so the area of drug development is getting very sophisticated in terms of how people are delivering these drugs to different systems, whether it's the surface of the cell, inside the cell, or targeting, say, the Golgi complex or mitochondria, or even on the outside of the nucleus to affect gene transcription. So this is the story so far. They're cannabinoid receptors, and they're just about everywhere. The most abundant protein in the mammalian brain. So these receptors have a certain structure. They're what's called a 7 transmembrane helices. So you have areas kind of like a snake in the grass, and you have amino acids that are important. And we've studied the importance of these amino acids. And one of the things that we didn't have when I was in grad school was a crystal structure of the receptor. We had to go out and create our own back in my day. And so we did that by doing point mutation analysis. And we mutated different amino acids to help understand how the receptor worked. Why would it recognize certain drugs and why others? But we also, while doing this research, started to find that if we changed certain specific amino acids in this protein known as the cannabinoid type 1 receptor, anandamide, the endogenous, the naturally made compound, would stop working at the receptor. THC might stop working at the receptor, but other synthetic compounds would still stimulate the receptor. Or if you added another compound, it would help to change the receptor's function, modulate it to help drugs bind to it. Fascinating stuff. And so when we talk about this receptor, we're talking about um, in a lab mutating it to alter its activity. And why is that important? Well, when we normally think up here on the top, we can see anandamide, and to the right, THC. When they stimulate these receptors, it normally initiates what's called a G protein coupled cascade, something we've published about extensively. Basically, this is normally what happens, the cannabinoid receptor, and there's downstream cellular responses, and the cell sort of goes about its business of being stimulated by a cannabinoid. And this is a cycle that repeats. However, the signal can actually change depending on what you're putting on the receptor, as well as the state of the receptor. So in an article where we developed some of the nomenclature around cannabinoid receptor pharmacology, one of the things I'd like to draw your attention to is the concept of allosteric modulators. And spoiler alert, this is how cannabidiol or CBD actually interacts with the cannabinoid receptor. It's a negative allosteric modulator, or a NAM. That is, it doesn't stimulate the receptor, it doesn't block the receptor, it just makes it less efficient at what it does. So if you think of the cannabinoid receptor as a volume knob that goes up to 10, CBD makes it so you can't go past 5 or 7 or something like that. You definitely can't go full throttle. So what does that look like? So if we talk about a positive allosteric modulator, something that would enhance receptor activity, we would expect it to be, much like a baseball player, right? His activity is pretty enhanced, diving for that ball. For a negative allosteric modulator, there'd be a decreased level of efficiency in terms of their ability to get the drug into the pocket. And that's different from blocking the receptor, which basically the receptor doesn't do anything and the drug just floats on by. So why do we care about being able to manipulate these receptors? Well, as I said before, there are mutations in these receptors, both in humans and rodents, that are associated with some really interesting things, like developing a substance use disorder, Parkinson's disease, your working memory performance, substance use disorders. One of my most fascinating reports, I think, that was ever published was looking at genetic mutations in anorexia and bulimia patients, folks with eating disorders. And what was found is that there was a population that actually had mutations in their cannabinoid receptor, their type 1 receptor, the one in the brain, the one that's THC stimulates, and the one that is responsible for appetite, and also, it also helps prevent vomiting and things like that. So it turns out that some of these was hypothesized by these authors that one of the reasons someone might develop an anxiety or eating disorder might be related to a dysfunctional cannabinoid receptor system, that they're not getting the signals to tell them they're hungry. They're not getting the signals to tell them, help them not throw up, as it were. And again, remember in 1985, the FDA approved purified THC for the treatment of chemotherapy-induced nausea and vomiting. So I think it's a fascinating aspect. And it led to a paper by my colleague, Ethan Russo, on what's called the clinical endocannabinoid deficiency syndrome, which is explaining difficult to treat symptoms as a disruption in this signaling system that is so pervasive through our body. So what another aspect to consider as well is that this may sound all fun and dandy, but this is big business. These are some funded clinical trials, both public and private, that are currently underway. You don't need to know everything about it. This is just to say people take this seriously and are actually using different genes to target populations. Now, let's try to tie it all together. So you have an endocannabinoid system. So I'm going to tell you about a case report. This is a patient who said, morphine has no effect, does not relieve my pain. This patient had a history of non-serious injuries, never needed pain medication, not even after surgery. High threshold for pain. How could the endocannabinoid system explain this curious case report? Well, it turns out that this patient had a mutation in a gene called FA. What does FA do, you ask? Great question. It degrades the endogenous cannabinoids, sort of recycles them. So your body has an experience, it produces endocannabinoids like anandamide. FA normally chews it up and gets rid of it. They're short acting. However, in this patient who had a FA pseudogene, which is it, they had the gene, it just was not functional, they had very high circulating levels of anandamide. Ananda is the Sanskrit word for bliss. So the endogenous cannabis compound is literally called the bliss compound. And if you exercise today, you probably felt the effects of anandamide, or if you got really hungry. But I thought it was very fascinating that in a history of painless injuries, the 66-year-old female was found to actually have an endocannabinoid mutation, which led researchers to further hypothesize that, what's really interesting about FA is that's also the target of Tylenol. When Tylenol is metabolized, one of the things it turns into is a FA inhibitor to allow the endocannabinoids to increase and help control pain and inflammation. If we didn't have an endocannabinoid system, Tylenol would probably have no effect on pain relief or inflammation. So next up, we're going to start diving into the more experimental area. I'm going to dive into a little more detail about THC, CBD, and these fun TRPV1 receptors. We'll talk about approved drugs, drug combinations, and the opioid epidemic. Some of this stuff we're going to go quickly. You don't need to memorize everything on these slides. That's just sort of put there to cover some bases for some of the things I'm going to say. So THC, a very well-studied cannabinoid, first isolated and identified in 1964, positively identified, that is. It was actually initially isolated in the United States, but because of quality control issues, they didn't realize it was the active ingredient. They thought the degradation component was actually the active ingredient for a long time. But it was in Israel that this was determined. So it has pain relieving effects. It's also a bronchodilatory. If you've seen the movie The Motorcycle Diaries, Che Guevara uses cannabis to treat asthma. We wouldn't recommend that today, but I think that speaks to its bronchodilatory effects. It is a very powerful anti-inflammatory agent. It's been compared to hydrocortisone. It's a muscle relaxant, has antiemetic properties as previously spoken. And it's also been shown to decrease beta amyloid plaques. It has, also it should be mentioned that THC, CBD, these don't have toxicity on healthy cells. They don't actually kill brain cells. It's a bit of a myth. They have their own risks unrelated to, they do not harm healthy cells. So cannabidiol, or CBD, was isolated by an American chemist first, Roger Adams, a very famous chemist. It really doesn't bind or stimulate CB1 receptor. It works as a negative allosteric modulator. It inhibits anandamide uptake. That is, it helps to increase it. It's alerting versus THC. It does not typically cause sedation. It has notable anticonvulsant properties, hence why it was Schedule 5 in the form of Epidiolex, which is an anti-epileptic medication. It has some documented anti-anxiety effects. It has shown some remarkable effects in breast cancer cell lines, that is in petri dishes, and it stimulates receptors called orphan receptors. That is, there are these receptors, we really don't know what they do, but turns out CBD stimulates with them, which is a fascinating aspect because we're starting to figure out what these other receptors do that we don't understand because of CBD. It's really kind of, everyone likes to talk about the ancient use of cannabis and how that's why people should use it. But for me, I don't think that's such a great rationale. We've also used natural products to kill each other for thousands of years. And so I think what's fascinating is the modern research today is what's promising, more so than ancient use. And again, enhancing adenosine signaling activity of the 5-HT1A receptor speaks to its ability to affect mood and energy. It also has been shown to prevent prion accumulation associated with mad cow disease, for example, and also to stimulate bone healing in animal models. So one of the ways that cannabidiol exerts its effects is through this TRPV1 channel. This is an amazing receptor. It's an ionotropic cannabinoid receptor. It does a lot of cool things. It senses temperature, mechanical and osmotic stimulation, such as swelling, senses things like electrical charge, light, chemical stimulation. And you might have experienced this today if you ate a chili pepper. So capsaicin stimulates the TRPV1 receptor, which sends this little biting pain to your brain. CBD can actually desensitize that receptor. I don't recommend you try this at home, kids, but if you took CBD oil and coated your tongue with it, it might actually block the effects of the chili pepper. And while you may not feel it in your mouth, there's no guarantee it won't protect the rest of your GI system. So, be careful when trying to show off this trick. So we've talked a lot about cannabinoids, THC, CBD. There's 100 other that have been discovered on the plant, but there's another aspect of cannabis that is really important, and that is the terpenes. Now, these are what give cannabis its fragrance. THC, CBD, they don't have a smell. What has a smell are these essential oil compounds found on cannabis, which are also found on many, many other plants. Limonene, for example, found in the rind of lemons, it actually counteracts some of the effects of THC via the 5-HT1A receptor, similar to CBD. And this is partially maybe help, plants, products with a high amount of limonene might actually have less anxiety sort of provoking properties. Pinene, for example, is an interesting compound. And if you know your Shakespeare, you will know that it is in rosemary and the herb of remembrance, which is a Hamlet reference for those of you who aren't up to date on Shakespeare. But it's an acetylcholinesterase inhibitor. It helps aid in memory and short-term memory recall. And this combined with THC can be quite stimulating. So if you were to consume a cannabis product that had a high amount of pinene, such as through inhalation, it might actually keep your mind buzzing all night. Not a great idea to consume THC and CBD at night. However, myrcene, also found in hops, which is the closest living relative to cannabis, actually at one time, 40 million years ago, give or take 100 million years, hops and cannabis were the same plant. And one day in Central Asia, they just sort of separated and became two separate plants. And so hops actually produces a lot of myrcene, which we know is a sedating muscle relaxant. Linalool, found in lavender, anti-anxiety compound, topical anesthetic, very interesting. So, this pharmacology has not escaped GW Pharma and other private companies who leverage this in their products because they want to mitigate, they want to tame the effects of THC. A couple other high notes here: beta-crofelein, crofelein oxide. These are ubiquitous terpenes found in nature, and they also stimulate the type 2 receptor. And if you ate a salad today, you probably ate about 200 milligrams of beta-crofelein. Actually, beta-crofelein is one of the major factors in the Mediterranean diet, also found in cannabis. You don't have to get it through cannabis, but it is an amazing compound. It's actually been shown to treat cocaine addiction in rats. So if you have any rats that are struggling, give them some beta-crofelein. What's also interesting is that the crofelein species is also what law enforcement has used historically to train drug sniffing dogs. Why they chose a compound ubiquitous in nature to train drug sniffing dogs can only be answered by those law enforcement agencies. So great. We have this pharmacology. We have this detailed aspects of where these cannabinoids come from, where they go in the body, and how that system works. How can we use it? Well, we have three flavors of cannabinoids. We have the endogenous ones made by mammals like us, anandamide and 2-AG. We have phytocannabinoids that are made by plants, cannabis sativa being the most famous, but also a couple of other species make cannabinoids, different varieties. No other plant makes THC or CBD, but cannabis is much more than just THC and CBD. There are many other alphabet soup compounds like cannabichromene or CBC, which occurs in rhododendron. But don't worry, rhododendrons haven't been added to the DEA schedule watch list yet. There's also synthetic cannabinoids, such as those made in a lab, FDA-approved Marinol. You can see here Marinol in the bluish thing with the, I believe, the 2.5, 5, and 10 milligram versions of it. I like to refer to it as the caviar of prescription medicine. One, because it looks like caviar, and two, because it's very expensive. Here's a fun fact. Again, another great cocktail party question, or tea party question. Where in drug scheduling is THC? Is it Schedule 1, the most restrictive category? Is it Schedule 2, the slightly less restrictive category? Is it Schedule 3, where it can be prescribed off-label? The answer is all three. THC in the cannabis plant, as we know it in the state programs and licensed operators, is a Schedule 1 drug. If it's in Syndros's formulation here in the middle of the screen, a liquid suspension of THC, it is a Schedule 2 drug. If it's in the form of Marinol, it is a Schedule 3 drug. But if it's in the form of flower, it's a Schedule 1 drug. Don't ask me why the same compound appears in 3 different scheduling categories and what the rationale is. Most notably, this is cannabis floss here. This is actually flowers from a Dutch pharmaceutical company. Their cannabis is gamma irradiated and goes through all sorts of processes, but meets pharmaceutical standards and is distributed through pharmacies and reimbursed by insurance in places like Germany and Italy, as well as Holland. Sativex, which is oral mucosal spray, there are something like 10,000 patient years of data. This is the most widely studied extract. It's approved in over 30 countries, but it's been in phase 1 studies in the United States since 2001 or so, not getting approval. However, Epidiolex from the same company, GW, has recently, again, gotten approved to Schedule 5 and then removed from the scheduling category altogether, and is again used to treat pediatric epilepsy. So we have some flavors, right? We have these phytocannabinoids and synthetic cannabinoids. And one other thing about Epidiolex I'd like to mention, the CBD medication, is that it is purified from cannabis plants. So you see that these are a variety of formulations. You see there is a pill, there is a spray under the tongue that's absorbed buccally. There are inhalable products, and they all have their pluses and minuses. So if you're looking at smoking or an inhalation or vaporization, it's a quick onset with a duration of about 2 to 4 hours. Now, it's great. It's rapid action, great for acute pain. Glaucoma patients report liking to use inhalation because they can help to control flare-ups, right? They need immediate relief. The issue is that there's some dexterity required. Vaporizers are expensive. Not all of them are portable, and not all of them are created equal. Oral administration, slightly problematic. Takes anywhere from 1 to 3 hours to kick in and can last upwards of 6 hours. But, it doesn't stink. No one's going to know really that you're consuming it, much like inhalation. However, there are titration challenges to using it. There's oral mucosal sprays like Sativex, which are sprayed under the tongue. It's not approved in this country, but it's approved in over 30 other countries, including Canada and Mexico, for the treatment of multiple sclerosis. This is sprayed under the tongue, absorbed buccally. It goes right to the brain. What's fascinating about Sativex is if you take a product and it's absorbed under the tongue, it's not detected in the blood like oral and inhalation. So if you got stopped for roadside sobriety testing using Sativex, you'd sail right through it. Whereas if you had inhaled the same product, you would fail it. Presents some challenges for developing those roadside sobriety tests. Again, this is more information here going into more detail on routes of administration. So I'm going to skip this and move on in the interest of time. So you may be thinking, well, these things sound pretty good. There's science behind them. There's these different administration forms. Maybe we should think about prescription pills and CBD. Well, we're going to get into that. But first, I'm going to talk to you a little bit about opioids and cannabis. So this has been a big topic, and I think it's worth discussing cannabis and opioids because this is a big contentious issue about, does cannabis reduce opioid use? Does CBD reduce opioid use? Does it reduce our reliance on it? Well, opium has been a popular substance since about 1500 BC. In the 1880s, the East India Company, the largest corporation ever to existed on the planet, was shipping about 80,000 chests of opium a year to China. That's about more than 4 million kilos. It's estimated in the first year of shipping opium, they created over 12 million addicts. And again, opioids act through receptors, much like cannabinoids. The mu opioid receptors associated most with the pain relieving effects and respiratory depression. And again, the word endorphins comes from the term endogenous morphine. And we have a lot of different types of opioids: morphine, codeine, fentanyl, and tramadol. And don't worry, we don't really need the plant that much anymore. Scientists have figured out how to grow it in yeast and to produce them in mass. So I think it's important to also think about drugs that are being marketed today and drugs that have been marketed in the past. So maybe your great grandparents may have had Bayer Pharmaceuticals heroin, or Miss Winslow's soothing syrup, which I found this delightful op-ed in the archives where they described it as, like magic, he soon went to sleep and all pain and nervousness disappeared. Great for teething children. Also this laudanum that I guess is geared towards sailors, and also an ad from a magazine for Quaaludes. And cannabis was not immune to the this lack of empirical approach to medicine. This is a bottle from the late 1800s, which contains alcohol, morphine, cannabis, and chloroform. It was prescribed to treat headaches, which is great because it would be the last headache you ever had if you took that. So cannabis and opioids have been combined for a while. Here are some examples of the common morphine products. So fentanyl is a really dangerous substance. It's 800 to 1,000 times more potent than morphine. On the right, you can see a visual representation of the lethal dose, of which 50% of the rats in the study would die of heroin. And that on the right is fentanyl. And then on the bottom, that is the lethal dose of cannabis. That is, if it was dropped on your head from a large height, it would kill you instantly. That is because cannabis has no achievable lethal dose through normal administration routes, inhalation or eating it. It simply does not cause a toxic, lethal overdose. But fentanyl is a medicine. It is used in transdermal patches. There are fentanyl lollipops for people undergoing cancer treatment who can't swallow and their throat becomes swelled up. So it does have its uses, but it's been diverted into street drug formulations and has found its way into a lot of substances. So researchers have actually started to look at CBD and fentanyl and found that CBD co-administered with opioids was safe and well tolerated, which provided the foundation for future studies looking at CBD as a potential treatment for opioid abuse. So what do we know right now about cannabis and the opioid epidemic? Well, research has shown that there is not an increased drug abuse liability when co-administered with opioids. I know it's hard to believe. It's been linked to lower numbers of opioid prescriptions. It's been linked to lower numbers of overdose deaths. However, this data is highly debated because in some states, they didn't see a decrease in opioid overdoses, but in those states, they have different programs. So some states have adult use programs where anyone can walk in and buy cannabis. Others are much more restrictive in terms of the products they have available. And there's also been a decrease in opiate positive screens in fatal car accidents recently. So what about the mechanistic proof? Animals deficient in cannabinoid receptors actually have reduced opioid reward. And what's interesting is that opioids regulate the release of endocannabinoids, and endocannabinoids alter our natural opioid levels, that is our endorphins. So these two systems talk to each other and help to regulate each other. The runner's high that we think about is not just endorphins. It's actually endocannabinoids that are leading that charge for runner's high. Endocannabinoids and opioid receptors, they're expressed in the same cells in discrete neuronal pathways, and they share similar intracellular signaling cascades. So the point is that they converge on each other, these endogenous symptoms. And this allows researchers to exploit, looking at opioid use disorder and opioid use public health aspects of these drugs. So let's talk about the data, and then we'll go into some clinical research. So cells and receptors, right? So we know that CBD can allosterically modulate mu and kappa opioid receptors, much like it can modulate CB1 receptors. And in animals, CBD can reduce the rewarding properties, attenuate Q-induced heroin seeking, as well as interact in important areas of the amygdala. In humans, modulating anxiety and those triggers for addiction and relapse, reducing amygdala activation, which is associated with stress-induced relapse. And a pilot study showed that CBD reduced heroin-related Q-induced cravings in heroin abusers. And this translates into some serious outcomes. Questions remain, however, about its efficacy at different phases of the abuse cycle, initiation, relapse, maintenance, this sort of thing. And what's interesting is, unlike most opioid medications, there's a low value on the illicit market for standardized cannabis preparations. Marinol, for example, Epidiolex, does not have a huge resale value, and reports of diversion are very minimal. So my colleague and I thought, well, that's interesting. What phase of the abuse cycle might CBD be good for? So we authored a case report where we tracked the use of acute opioid withdrawal in a patient who was going off a pharmacological treatment known as methadone. And basically, this was the first of our knowledge of a case report using CBD during the transition to abstinence maintenance therapy for opioid dependency. And what was really interesting is, an 8-year history of severe opioid use disorder takes this naltrexone and methadone-based abstinence. CBD seemed to help him with that transition. We also looked at another case of a comorbid symptom, and this was published in the American Journal of Endocannabinoid Medicine. Looked at rheumatoid arthritis, lupus, and Raynaud's disease, 3 difficult to treat conditions, tracking a patient using 300 to 600 milligrams per dose. It seemed to help, and this was an extract that helped manage complex pain symptoms. So these are case reports. Some people would say they're anecdotal. Some people would say they're the foundation of future research. I mean, after all, Epidiolex's emergency status what started off as a case report in a young girl suffering from pediatric epilepsy. Knowing that this data wasn't good enough, my colleagues and I in Delaware decided to take it a step further. And so we tracked 80 cannabis patients and their opioid use for 6 months. And we tracked neck and back pain. And these are all patients who had morphine milligram equivalents, as they're taking opioid medication. And we track them at time 0. The day they become a cannabis patient, certified and able to buy illegal cannabis in the state of Delaware, for 6 months. And we found an average of over 30% decrease in opioid use. Again, this is in a petri dish, and that's about the size of Delaware, but I still think that this research is promising, given that there are more than 20 pain studies currently going on at clinicaltrials.gov. So I'm going to move through these next slides a little bit quickly in the interest of time. I know we have some questions that I'm sure you guys would love to ask, but I want to talk a little bit about some of the effects of CBD, notably the side effects of CBD, and spend a little attention to drug-drug interactions. So just quickly, again, we have THC and CBD. The sensitivity to both is highly variable. What's an efficacious dose in one person? Not tolerated in another. It requires a trial and error approach. So drug-drug interactions, or DDIs, right? These occur if you're taking more than one medication, right? And the reports of adverse events of multiple drugs is rising in the United States. And we're going to focus on one aspect of this. And we're going to be looking at the pharmacokinetic interactions. We're going to explore cannabinoids really briefly as victims, where the THC or CBD is changed, the levels in the body are changed by another drug, and where cannabinoids are the perpetrator, where they cause the change of another drug. And there might be times when there are overlapping side effects, where THC causes sedation, you take it with another drug that causes sedation. That's a pharmacodynamic effect. The idea of victim and perpetrator don't really apply there. Let's focus on drug metabolizing enzymes, cytochrome P450, the CYP molecules. Um, there's a huge variability in this among people, hence this pie chart. And this leads to a lot of drug-drug interactions, as some people are slow metabolizers, ultra slow metabolizers. Some people are fast, some people are ultra fast. There's actually in sickle cell anemia populations, they are ultra rapid metabolizers of opioids, as they can take an opioid and not even feel it. It's so fast in terms of its metabolism. And similar for cannabinoids, there are people who walk among us who are exposed to a cannabinoid and don't feel as much of effect as some other people who are slow metabolizers. So what does this look like? The bar graph might explain it best. So when you look at cannabinoids as victims of drug-drug interactions and look at it with a CYP3A4 inhibitor, that is something like chlorothromycin or ketoconazole or some sort of protease inhibitor, in the blood, you'll see 2 to 3.5 fold increases in THC and its metabolites in the blood. So that edible you ate might pack an extra wallop, as it were, because it's not going to be metabolized as quickly. Similarly, cannabinoids can be perpetrators of drug-drug interactions. Clobazam, or a metabolite of it, it's a benzodiazepine. When co-administered, there's a higher amount of it, for example, when co-administered with CBD, and this has been observed in clinical trials, especially in epileptic patients. So CBD can have a number of drug-drug interactions. These are generally in the category of proceed with extra precaution. And that's because CYP3A4 is a common target. I think something like 60% or so of clinically prescribed drugs are metabolized through the same CYP, which could lead to some potential issues. Side effects of CBD, no one really likes to talk about this stuff. In animal studies, there's been some, again, in animal studies, the purpose is to give the animal as much as possible. In toxicology studies, to find where the harmful effect is, not that it has relevance to human consumption per se, but they can see a decrease in cell viability, reduced fertilization capacity in mice and rats when administered superhuman doses of CBD. In humans, a CBD is associated in chronic dosing, dry mouth, nausea, diarrhea at exceptionally high doses, potentially increasing intraocular pressure, although this is not confirmed. The effects on hormone levels are also unknown, and there are drug-drug interactions. Let's be fair. Let's talk about THC and Marinol. There are common and uncommon and rare effects of it. Very common, drowsiness, fatigue, dizziness, dry mouth, acute anxiety. Sometimes there can be cognitive effects, but things like blurred vision, headaches, or depression or tachycardia, these things are very rare in terms of cannabis or THC's effects. So we're going to round out this presentation talking about products, particularly labeling and quality of products. So what's in a label? Good question that someone asked. And in a study, only 5 out of 48 participants could identify what the universal symbol for cannabis meant. So here are from 7 US jurisdictions, the universal symbols for a THC containing product. As you know, regulators are using a definition of universal that I am unfamiliar with, as they are all different. But participants actually had a misperception about what it actually meant. And so we have this issue where people aren't sure what it meant, because hemp products do not have this warning label, even though if you have a chocolate bar, if you have let's just take a 5 gram gummy bear could have upwards of 10 milligrams of THC in it, because it meets that 0.3% definition. A 28 gram or a large chocolate bar could have 1,000 milligrams of THC and be illegal. So be careful with those hemp products, as they do not carry this seal of notifying you that they actually have THC in them. One of our most well-cited and popular articles is our Journal of American Medical Association study on the accuracy of CBD products. So we purchased or so products online and had them tested by 2 independent laboratories with accredited methodology. And we found that 69% of the products were not within 10% of their labeled accuracy. That is, basically 70% of the products on the market are not accurately labeled. So just remember what Abraham Lincoln said when he posted this selfie. The problem with CBD on the internet is that you can't always depend on the authenticity. So, and I'm happy to share these slides as I know I'm going quickly through them. Things to look for a product: tamper evident, child resistant, resealable, non-transparent. A product identity, a universal symbol is mentioned, a net weight, the servings and milligrams of THC and CBD, as well as an expiration date. I think that's an important thing on a product. Manufacturer's name and contact, lists of ingredients, instructions for use, batch numbers, as well as any sugar, sodium, carbohydrates, allergens, and artificial food coloring, if applicable. So some states, like the Washington State Liquor and Cannabis Board, actually define edibles and cannabis products. They say you only can use dull colored candy. You can't use bright colored. Here are approved shapes, as well as your swatch of approved colors for packaging of the product. And how can you tell if a product is a cannabis product versus a hemp product? Because in the hemp industry, there are no rules. As you can see, this is a tinfoil psychedelic packaging that contains whatever 99.9% CBD crystals are. I don't know. I've been in the field for almost 20 years, and I don't even know what that means. But when you add up the ingredients, you find that there's 400 milligrams missing from the added up ingredients, which is kind of alarming because there's supposedly only 100 milligrams of CBD in the entire package. And that's why you always want to buy from licensed operators and not the gas station or Bed Bath and Beyond or whatever, because these products are this product is absolutely not regulated, has escaped any testing or qualification to be on the market. Unlike products which follow this document that me and many other people have co-authored that was adopted by states, helping to set these standards of identity analysis and quality control. So there are standards out there to help guide cannabis and hemp products that are through licensed operators. And I wrote about this for Nature magazine. And I think the best thing that I said in that entire op-ed was that the priority for the US government should be to facilitate research which will help inform our education and policy agendas. If we don't want kids to use these drugs, we should really research what works. If we want to have safe products, we should research what works and have more informed education and policy agendas versus just what sort of sounds good on a soundbite. So in summary, in the 1990s, a new signaling system was discovered, known as the endocannabinoid system, which turned out to be the most abundant protein in the mammalian brain. That's like living in a house your whole life, and then one day you discover that there are light switches in every room. What does this system do? It helps you to eat, sleep, relax, forget, and protect. CBD is probably more effective as an extract than as an isolated agent. It does have its limits. It has potential side effects and potential drug-drug interactions. We should use science to inform our regulations, industry, and research. If you're looking at buying a product, if an employee or there is not a pharmacist or health professional available that can explain the product clearly, consider choosing another product or location. And remember to be safe, buy from licensed cannabis and hemp operators. And if you're interested in trying these products, the old advice is to go slow and stay low in what you're using. So again, be careful if you're thinking about trying edibles. Do not underestimate them. All right, I know we went over time just a little bit. I want to thank you. If you enjoyed this, I'd encourage you to check out my podcast that me and other people with letters after our name put out every 2 weeks called How to Launch an Industry, where we discuss the science of psychedelics and cannabis and other drugs of abuse. We just put out a new episode called From Indigenous to FDA, where we talk about how substances come out of indigenous populations and into the FDA pipeline. And with that, I'm happy to take any and all questions loosely related to cannabis. Thank you. All right.
There is a question that came in on the chat. I'll read the question real quickly. It came from Carol Chacon. Well, actually, that's not the question that I wanted to read. They're scrolling by me too fast. From Shirley Wendell. How do you know if a seller is licensed?
That's a terrific question. If you want to know if a seller is licensed, you ask them for it. It might be on the wall somewhere, and it's something they would probably brag about. Rite Aid is not a licensed cannabis operator. The gas station is not a licensed cannabis operator. These are typically very specific, focused operations that sell exclusively cannabis products. So again, I think it's fair to ask them, are you a licensed cannabis operator? Do you have another great question to ask that is ask for testing records of these products. So every product should have a certificate of analysis that is available on file. So you can ask for the testing records of those products. If they can't provide it for you, or at least send you to a link or a website that has information, I would not ingest that product. So I would just ask them if you are unsure.
Next question is from Lauren Eisen. She's asked topical CBD oil with THC, and then i.e. 5:1 or 1:5. How does that work? What to look forward in best quality?
Yeah, great question. So, there's an old joke that CBD topical oils work less than our last president. But when it comes to topical application of CBD oil, the skin is the largest organ we have. It's not just some, barrier that's inactive. There are receptors on your skin surface. And so you have actually cannabinoid receptors in the follicles where hairs are coming out with and things like that. And so if you have psoriasis or a skin issue, cannabinoids may be helpful. You have to kind of be careful with some of these preparations though, because some of them are great at treating acne. Some are great at moisturizing the skin. No product can do both, because if you're moisturizing the skin, you can't dry out the sebaceous glands and treat the acne. So again, something to think about when you look at these products and help calibrate your baloney detector. If it treats something or claims to treat something like acne, it can't also moisturize the skin. Those are going in opposite directions. So how does it work? Well, topically, it stays on the skin and it works there. If it's penetrating, it can go beneath the skin, but this is very difficult to do because under our skin, we have a layer of fat, and cannabinoids are fat-like compounds that get stuck in that fatty layer. Actually, your body loves to just suck it into fat when you consume it orally, because that's why you can fail a drug test like 30 days after using cannabis, because it's sucked into your fat and slowly released out, because your body thinks it's fat. So topical application has a specific mechanism on the organ known as skin, where you have receptors. So it's a great question, I think. And when you look for quality, look for consistency in labeling. Like I said, if it's a topical application, it shouldn't also be penetrating. If it treats acne, it shouldn't also be moisturizing. You see a lot of weird claims about these products. Again, look for a good manufacturing practices, a GMP seal or something like that on these products. A third-party certification.
This is Pete. Excuse me interrupting for a second. Shirley Windle, you're sharing your screen. Is there a reason for that?
For what?
Okay.
While they're figuring that out, Dr. Jane Patton asked the question, to what degree are MDs up to date?
Oh, well, that's a great question.
They're
So
all great questions.
to what degree are MDs up to date? The only MDs that are up to date are the ones that have taken it upon themselves to research this. And to, again, the first CME, the first continuing medical education available to clinicians on this topic, was approved, I believe, in 2001. And medical schools still do not teach anything other than you have cannabinoid receptors. And that's about where the medical school lecture typically ends, if that at all. So I think it is a big issue that we don't really teach clinicians about the most abundant receptor signaling system in the mammalian brain. So, and then you add on top of that the explosion of product diversity that is happening right now in the United States. Clinicians are actually relying on you to help educate them and inform them. And many times I give patients and friends research papers to bring to their clinicians and to be able to more have an adult conversation about cannabis. And you have to be clear when you're talking with a clinician, if you're talking about street marijuana, a drug that can be any green leafy vegetable material used to get high, versus a medical cannabis product, which has a defined identity and characteristics and is a legally regulated product. Those are two completely different things. And it's important to distinguish that. So some doctors are up to date, many are not. And I think that's unfortunate right now, but we're, a lot of people working to change that. The journal that I'm the editor in chief of, we actually target clinicians for our publication and spend a lot of time trying to translate and focus on articles that have a translational potential to the clinic.
Next question is from Linda David.
This was so much information. Is there a way that we can replay this?
Invite me back.
Hold on a second. This is Pete. This will be up on the resident website together with recordings of all other community forums. So in about a couple of days' time, maybe tomorrow, you'll be able to view this entire presentation.
Great, thank you.
Can I ask you, can cannabis be used for either essential tremors or Parkinson's disease?
That is an active area of research. In terms of the best evidence, they've looked at different neuromuscular disorders and things like Lou Gehrig's disease and multiple sclerosis. Cannabinoids seem quite remarkable in that regard to be able to control those symptoms, especially in multiple sclerosis related tremors and spasticity. It's one of the best studied applications of cannabis extracts, in my opinion. But as far as Parkinson's disease goes out, I think there's something like 3 small clinical studies exploring this. And so the jury is really out on that, but it is an area of interest because it does appear that cannabinoids can help control the tremors. But there are other issues associated with Parkinson's disease that make it difficult to treat. But again, I'd expect there to be products developed for this in the near future due to the success of Epidiolex or the CBD preparation to treat spasticity, convulsions, and tremors associated with seizure disorders, as well as the cannabis extract Sativex. That's not available in the US, but in 30 some odd other countries is a licensed pharmaceutical used to treat muscle spasticity and tremors. So I think there isn't a lot of good evidence for Parkinson's disease specifically, but for neuromuscular conditions, absolutely. And one of the things I'd like to remind people is that cannabinoids are prescribed to treat neurological conditions, not to cause them. And I think that's one of the great promises that these compounds hold, is for treating neuromuscular and neurological disorders.
That's all the questions that we've got recorded thus far.
There's one over in chat.
Let me go to the chat box.
Scroll up. No, still gotta keep going.
No, I got
Wait.
it.
Bob, this is Pete. Laura Eason has her hand up.
Yeah, I've tried to unmute Laura a couple of times.
Oh, am I there?
She's unmuted now.
Okay, so I was the one who had asked about the, excuse me, the 1 to 5 or 5 to 1. I can never remember which way it goes. CBD with THC, primarily for pain, not skin but for
Mm.
inflammation, joint inflammation, things like that. I wasn't sure exactly how that works. I know it's working by being absorbed into the skin, but I'm not really understanding what the 5-to-1 or 1-to-5 represents, and not exactly sure how that's working with the pain receptors.
So I'm going to take a stab and say this product. Uh, so one of the things that's really remarkable about cannabis and its products, hemp products, is that how it's escaped clinical approval. Normally, to put a drug out there, it has to go through a clinical study and all sorts of regulatory approval. Not for these products. And so chances are high in the United States that if you're getting a product that has a ratio on it, that product has not been tested in animals or human beings. They're probably basing it on some, PhD's thesis research, and they're loosely associating it to their product. So again, what does the 1-to-5 ratio do? Well, there's a rationale there, a speculative rationale, but the most important ratio is 1-to-1, 1 THC to 1 THC. And the reason is because if you consume that orally, for example, or under the tongue, it will block the intoxicating properties of THC, that CBD. Now, why would you want to play with that ratio? Because you may want to enhance or inhibit certain activities of, say, THC. Maybe you want to take the edge off it. Maybe you want to enhance the pain relieving properties. But again, many of these companies have not performed clinical research on these products or animal research. So
So if we're
again,
obtaining these products in a dispensary, then should we not presume that they are tested? Because that's where I was getting the topical CBD oil with the THC in it that was being used for pain by a family member topically.
yeah, so they are tested for purity, consistency, and safety. Like there's only, like for heavy metals, fungi, bacteria, and things like that. But in terms of clinical efficacy, that data is largely based indirectly studying other products or drugs. So that specific product probably has not been studied, maybe at best in an observational survey type platform, but I'm going to guess probably not. It doesn't mean does not have a therapeutic application. I'm just trying to sort of help you understand that they are the operators are trying to fill in the gaps because clinical research actually on cannabis isn't typically allowed. You're allowed to do a phase 1 study, but no phase 2, phase 3, or phase 4 study has ever been approved for a THC cannabis product. Because why would you do a phase 2 or phase 3 study to look at therapeutic benefits of a Schedule 1 narcotic? Because by definition of Schedule 1, it has no accepted medical value, so you can't get approval for a study looking at the medical value. And onward, this circular argument goes with the DEA and regulatory bodies. But some drugs like CBD, which does not cause a high, has managed to get into the FDA pipeline and be studied in phase 2, 3, 4, 5 clinical studies. Sorry, I know it's a bit of jargon there, but I think it's important to understand the environment that these operators are in. They don't have a lot of mechanisms to actually study these products in traditional pipelines, traditional formats, because of the federal illegality of these products. Remember, all cannabis products are Schedule 1. They're all federally illegal, hence why you can't take them across state lines, ship them across state lines, and you don't see cannabis dispensaries like McDonald's where the same one is operating in every state. And the cannabis isn't in this place where it's not like oranges, where you grow orange trees in the best place to grow orange trees and you make orange juice and ship it around. If you want cannabis in New York, you have to grow it in New York. If you want cannabis in California, you have to grow it in California. No other product in the world is quite like this. So it presents some challenges, making a multi-center clinical study just impossible to do, because how are you going to get it to the other center? So it does prevent some interesting hurdles to do research in terms of more what we might expect in modern day clinical studies. So I'm getting back to your thing. How does it work? I can't say specifically to that product without seeing it in more detail, but unless it's formulated to penetrate the skin, then it's probably just acting topically. It might be, there are other compounds like the terpenes that can act as local anesthetics to temporarily relieve pain, but cannabinoids can have an impact on rheumatoid arthritis. But in the recent article I published with my colleagues, there are huge gaps in our understanding exactly how these compounds work. There certainly is promise, and if you're a rat with arthritis, we could probably treat it really well. But there's some subtle differences between humans and rats, and making that leap is not quite there yet. So I guess is my roundabout way of saying yes and no. That is there a rationale, scientific rationale underpinning the therapeutic potential of using a ratio? Yes, you might want to enhance or inhibit certain effects of the drugs, but is there like clinical data to look at about that product? No. So I hope that helps.
It does. It really just tells me that it may have therapeutic value, but it will not be legitimized by the control standards that are in place right now.
Yeah, it's definitely a trial and error phase for patients. And it's said that the first month or two of being a cannabis patient is the most expensive part of being a cannabis patient, because you're trying so many products to go through a trial and error process to find what works and what doesn't work.
Well,
We
thank
have
you for
one
the
more
thorough
question.
explanation.
We have one
You're
more
welcome.
question, Doctor. This one has to do with smoking. And this comes from Bob Green. What other ways to take CBD or THC than smoking are effective?
Sure. So, well, if you have an orifice, you can put it in there. So there's lots of different ways to administer it. You can swallow it. You can put it under your tongue. There's transdermal patches, and there's also, suppositories are also quite popular as well. So you have a number of administration routes available. I think it's making sure you pick a product that's fit for the purpose you want to do it. So Epidiolex CBD, which is used again to treat epilepsy, is taken orally, about 300 milligrams per dose in the clinical studies. Other products like THC, maximum dose on THC for cannabis products, I think, is 10 milligrams per dose in the California market. So again, oral administration, I'm a big fan of the oral mucosal route, which is typically can be a lozenge, a tincture, a spray that's under the tongue. It's absorbed. Don't swallow it, because then you start to go through a different route of administration. But absorbing it buccally, I think, is a very efficient way because of its rapid onset and the ability to titrate the dose. So you have a variety of methods. Also, if you don't want to smoke, there are vaporizers and inhalation devices, and not vape pens, and I repeat, not things called vape pens. Those are like garbage toys from China. What I'm talking about are herbal vaporizers, and these are clinically developed devices for delivering like a steam or mist of cannabis. Volcano is the name of one company that produces something called a Vapor Medic, and these exquisite machines are like built in Germany in the same towns where they make like scalpels and stuff like that. It's a very sophisticated piece of equipment. PAX, I'm also on their health advisory board, makes a vaporizer that meets many different standards voluntarily that things like the FDA impose for inhaled devices. So there are lots of ways to ingest cannabinoids beyond smoking. There are acceptable routes of inhalation that are not as dangerous as smoking. There's oral mucosal, absorbed under the tongue, as well as transdermal patches and rectal suppositories.
I think we're up to the final question. And this comes from Shirley Wendell. There's two parts to it. In the arthritis study, how was the CBD administered? And the second part is, do you anticipate the federal legalization of cannabis and hemp?
For the first one, in the case report we published, I think it was 300 to 600 milligrams of CBD per dose. Quite a lot if you're buying, the sort of products you might find at a gas station or other stores. So 300 to 600 milligrams per dose. It's quite a lot. And this was in a complex, there were like 3 conditions going on at once. It's a very complex pain disorder. For the recent study we published, that was more of a review or op-ed style article. So again, but I hope that answers your question. Again, I'm happy to share the case report if anyone wants to follow up. Regarding the federal legalization of cannabis and hemp, I think that I don't have a crystal ball, but I would say that it is inevitable at this point. And I would say that even to this point, it is a bit of a dereliction of responsibility by federal agencies to not have already started to issue guidelines. I mean, how insane is it that they're leaving it up to states to determine what pesticides are safe to use on these products when we have a department, the USDA, which sets those exemption limits, but they can't do that because cannabis is federally illegal. I think that it is a big issue. So again, I would say that it is inevitable. I think that we have a false dichotomy right now, which is really a bad public health issue, and that's creating a difference between THC from hemp and THC from cannabis. One, you have to have a license to sell, the other you don't. It's created a bit of a serious public health issue, in my opinion. And more on that later. So I think with federal guidelines, we'd see things like interstate commerce. We would see more regulation, more studies, research and development would actually be reasonable to fund. And the most important issue, banking. Banking is a serious issue. And I'll share a personal story. Much like my family members, my bank also doesn't understand what I do for a living. And we actually had to change our bank. We lost our bank account. We lost our corporate bank account because we made a cannabis purchase, and it was for a research conference that had cannabis in the title. And the bank said, "Oh, you're a cannabis company." And they closed the bank account and sent us a check for our funds, and we had to find a new bank account, and quite a disaster. And so this is commonplace. And I don't even handle cannabis. I don't even touch the stuff. And I'm facing banking issues. I faced a banking issue. Dispensaries and operators. I talked to a colleague who works at a dispensary. Says, "Yeah, we have a 3-bank approach. We have one bank we use, one bank we're talking to, and a third one in backup, just in case when the bank is ready to cancel our account and for us to start over again." So cannabis is still federally illegal. And right now there's a case, I think, in Kentucky where a guy who's a licensed cannabis operator, because he couldn't get a bank account because the banks say it's federally illegal, lied about what he did. He said a wellness center. And now he's looking at money laundering charges because of that. So the banking issue is a serious issue in the cannabis industry. State chartered banks will allow you to have accounts, but again, it's a fascinating legal issue, but that has to be fixed before really anything can move forward in a significant way.
Okay. Shirley Schultz, do you want to say anything?
Yes, so I guess this is going to conclude our discussion for tonight, but Jehan, we sincerely appreciate your willingness to burn the midnight oil, where I guess you're in New York. And this has been a very informative session, and we hopefully may in the future ask you back. So thank you very much for speaking with us tonight.
It was my pleasure. A lot of great questions. I certainly had a great time talking about one of my favorite subjects. And in the future, I'd love to, if your community is interested in less technical presentations, there's some fantastic historical information and other explorations as well.
Okay.
And also, I have a survey that my colleagues and I are doing, a knowledge and attitude survey. So Shirley, I'll share that link with you. And if you guys would like to contribute to science, you can fill out the online survey.
Thank you very much. And if we want to be in touch with you, we use that email that you gave us.
Yes, or you can, my grandmother will also field all calls for me too. But yeah, so I will put that, I can put that in the chat right now. It's probably the best email, is the business one. If you have questions, comments, or again, if you're rewatching this and you want to, can I have a copy of that study? The answer is yes. So please feel free to reach out.
Okay, thank you.
Thank you. Good night.
All right, good night.
Good night, everybody.